right. Hello, everyone. I know it's early, but thank you all for being here. Welcome to the Bioinformatics and Computational Chemistry division of this conference. We have 4 presentations today, and at the end of each presentation, we'll take questions from the judges and then the audience if we have time. And you can ask your questions through chat or through the Zoom raise hand feature, and then wait to be called on. Before we begin the presentations, can the judges please briefly introduce themselves and mention their research field?
Sure, I suppose I can go first. My name is Luke Richardson. I'm a PhD student here at Baylor in Taraj Saluki's group. I work in the analytical chemistry division, so doing mostly instrumental analysis, some instrument modification, looking mostly at biological substrates and whatnot. So very happy to be here.
Thank you.
I guess I'll go next. My name is Evers Castaneda. I am currently in Dr. Eric Baker's lab. I am a graduate student in the cell molecular health and disease track. We mainly work on graph theory and looking at heterogeneous model organisms in, I guess, data analysis. Glad to be here. Hopefully I can see some really good experiments.
Thank you.
Okay, I can go next. Sorry, my video is not going to be on today. I have a baby in the house. My name is Patrick Olademehin, and I'm a 5th-year PhD student in Computation Chemistry Division. Presently, I'm working on molecular dynamic simulation, but I also have a project I'm doing on optimizing graphite carbonitrides as to make them better photocatalysts. So yeah, I'm happy to be here. Hoping to listen to great presentations today.
Thank you. All right, so those are our judges. Now we'll move on to the presentations. So first presentation we have today will be from Meg from Baylor University. And you may share your screen and begin when you're ready.
Perfect. Okay. I believe I should be able to. All right. Can everyone see that and everything?
Yes.
Perfect. All right. Well, my name is Meg McCutcheon. I'm a member of the Gallagher group here at Baylor. I will be presenting my research project titled An Application of Density Functional Theory for Characterizing the Energetics of Hydrogen-Deuterium Exchange of Solvated Glucose. So just to begin, in the Gallagher lab, we're really interested in understanding glycans. So what is a glycan? These are the sugar chains that are found on the outside surface of your cells. They're really important in signaling cascades. Because of the vast array of glycan structures, they're highly specific on your cells. So we hypothesize that by understanding the structures of these glycans, we can be able to compare things like cancerous versus non-cancerous glycan structures to understand some of the signaling cascades that are going on in those cells. But in order to really understand the structure of a glycan, we need to develop techniques in order to elucidate those structures. So why is this structural elucidation so difficult? Well, in sugars, we have a lack of functional group diversity, right? This image here shows that the most prominent functional group in a sugar is going to be a hydroxyl group. And then unlike proteins, which are, their sequences are determined by things like DNA, the branching structure of glycans is often determined by just the concentration of different sugars within the cell. So understanding these structures can be really difficult because of the large amount of branches and the lack of functional groups. So one technique that we use experimentally in order to study glycan structure is called electrospray ionization. So this image here demonstrates the process of electrospray. So our solvated molecule is sprayed using a spray tip in the Taylor cone. It's going to form a charged droplet, and as that droplet evaporates, our analyte becomes positively charged and enters the gas phase in order to move into the mass spectrometer. So we see that metals are what's charging these sugars during ESI. So other mass spectrometry processes involve either fragmenting the molecules or using other techniques in order to produce ions. So here we're preserving the conformation of these molecules, we're preserving their structures and adducting them to a metal in order to get those mass to charge ratios that we need in order to study mass specs. So specifically, we use an experimental technique called hydrogen deuterium exchange. We're actually labeling hydroxyls on these sugars. This image here is just demonstrating that we're swapping these hydrogens here for deuteriums. In order to do this swapping, we use a technique called theta tip ESI. So this theta tip is going to introduce deuterium at the same time that it introduces our analyte solvated in water. When we do this in the spray overlap region, the molecules are allowed to mix. But as we saw on the previous image, that droplet immediately evaporates. This is what's going to limit our time of exposure of D2O to the analyte molecule. This type of technique was first described for proteins. We see that proteins have very long exchange timescales, so it takes much more time for deuterium to exchange with some of those amino acids that are in deeper folds of the protein. But because sugars are so branched and hydroxyls exchange so rapidly, we needed to be able to introduce deuterium on the same time scale that we're having our analyte enter the mass spec. So that's where theta tips come in, is they're now on the same time scale so we can get differential labeling of these carbohydrates. So in order to study what's actually happening in these ESI droplets, we wanted to apply a computational technique. So the computational technique that I will be talking about today is called density functional theory. So this technique introduces the electrons as a probability function of space, thus eliminating the many-body problem. This is what allows us to optimize the probability function and calculate the ground state properties of the molecule. So specifically, my project is entirely computational, working to understand the energetics of NESI HDX. So part of my workflow is optimizing these structures using the Gaussian 16 platform. We then optimize these structures both pre and post exchange so that we can obtain the amount of energy that's released by hydrogen deuterium exchange. And then we compare across different parameters. So we vary the amounts of D2O in the first solvation shell. We vary the site of sodium adducts. Just to reiterate, those sodium adducts are what is allowing our sugar to be charged as we enter the mass spectrometer. And we vary the number of solvent molecules around our sugar, as well as the number of exchanges that have previously taken place on the sugar. All these experiments are done in replicates of 10, so error bars on all of the following graphs are going to refer to the standard deviation of those replicates. So the first thing that we demonstrated was just with a neutral sugar, showing that HDX is going to be an endothermic or an exothermic process, excuse me. So we showed that when this exchange takes place on these hydroxyls here, that's actually going to be associated with a release of energy. So this is a favorable process taking place in the mass spectrometer. The next thing we wanted to do was to explore the effect of a sodium adduct. So this image demonstrates that we have this sodium adduct on this secondary hydroxyl here on our model sugar beta glucose. We saw that this secondary hydroxyl was a much more exothermic exchange as compared to this primary hydroxyl. In fact, this primary hydroxyl actually shows endothermic exchange, so it's taking up energy in order to get this primary hydroxyl to exchange when its neighboring hydroxyl is adducted to a sodium. So from that, we learned that we had an effect both of the sodium itself adducting to the hydroxyl, but also to the hydroxyl's location. So we confirmed this by looking at now having that sodium adducted to the primary hydroxyl here. So this primary hydroxyl now is showing that larger release of energy as compared to the secondary hydroxyl, which doesn't have the sodium adduct. So again, we're seeing that the presence of that adduct is really stabilizing that exchange. So we wanted to propose a mechanism as to why the presence of this sodium was so significant in stabilizing exchange on these sugars. So research in a different group had shown that using IR spectroscopy, that when you have a metal present, you're actually getting some density withdrawn from this oxygen here. So this sodium metal in our mechanism is withdrawing electron density from our oxygen. So that adduct is actually making this hydrogen more labile so that we don't have such a great energy barrier for a molecule of D2O to come and remove that hydroxyl group so that it can be subsequently swapped for a molecule or for an atom of deuterium. So because of that sodium adduct, we actually have a lower energy barrier for exchange to take place, and that's why we're seeing a greater release of energy during hydrogen deuterium exchange. The next thing that we wanted to investigate was the effect of the solvation shell. So again, these droplets are forming in ESI and they're very rapidly evaporating. So previous experimental work in the Gallagher group had shown that hydrogen deuterium exchange is not going to take place once that sugar enters the gas phase. So we wanted to see, well, how large does the solvation shell have to be in order for these exchanges to be energetically favorable? So we confirmed here that when we only have one molecule, so we don't have a full solvation shell, HDX is going to require energy, so that's why we don't observe it experimentally. It's not favorable, but it actually takes a full solvation shell. So we see that once we get up to 9 molecules, that's when it starts to be an exothermic process. So without that full solvation shell of energy, HDX isn't going to be occurring. So this was a confirmation of some experimental work that we had previously seen in the Gallagher lab, but now confirmed with computation. So the final variable that I wanted to present to you today that we explored was the effect of multiple exchanges on a non-sodiated site. So when you have multiple exchanges occurring on this molecule, here we see that our sodium is adducted to this hydroxyl here, and then the site of previous exchange is on the opposite side of the molecule. So now we're exchanging this hydroxyl here. So when we're exchanging one that involves that sodium adduct, the sodium adduct is again stabilizing that exchange. So it's favorable for us to have subsequent exchanges. As shown by this graph, it's once again exothermic. However, when that exchange site is on the opposite side of the molecule, it's going to be more favorable. However, now that our sodium is adducted to our site that's now exchanging, again, we see that sodium is the greatest effector of what's going to stabilize the exchange of this molecule. So again, confirming what we had observed previously, when we're changing these other parameters, it is the sodium itself that's going to have the greatest effect on whether or not these exchanges are favorable. So here, this hydroxyl adjacent to this exchange site versus this exchange site, it doesn't matter where the previous exchange occurred. What matters is where the sodium is actually located. So here that difference was not significant. So once we had developed these methods, once we developed these applications for DFT using our model sugar, glucose, we wanted to explore larger sugars, specifically malesitose is the sugar that we work most often with in the Gallagher lab. And we saw consistent results here. So again, we have this much greater release of energy when we're looking at these adducted hydroxyls. So here this image maps out the different hydroxyls that are adducting to this sodium. This image shows sodium in the gas, or malesitose in the gas phase, but we again explored it with one full solvation shell around our molecule. And we saw that those adduction sites were having a much greater release of energy. We also wanted to compare the locations. So when we're working with secondary hydroxyls, we see that again, that adduction is having the greatest effect. So no matter primary or secondary, we had we were showing that, sorry, I was reading in the chat, yes. So no matter where that hydroxyl was located, the greatest effect was going to be whether or not sodium was there or not. There we go. So in conclusion, we were able to show that the exchange of secondary hydroxyls was more stable. That was consistent for both glucose and malositose. However, it is the effect of the sodium adduct that most greatly impacts whether or not our sugar is going to have favorable exchange. As it enters the gas phase, that exchange is no longer favorable. Multiple exchanges are going to be more favorable when they're occurring opposite previous exchanges, but the presence of a sodium adduct is more stabilizing than these effects. And these observations are consistent as we enter larger sugar systems. So thank you so much for your time. I would love to take any questions that you guys have for me.
All right, so if any of the judges have questions, you guys are free to go first. And while they're going, if anyone else in the audience has questions I'd like to put in the chat, they can.
Megan, that was a good presentation. It was easy to follow because you had it well outlined and you made your points very well. So basically what you said you're doing, you're just, you have hydrogen, then you have another system with deuterium. You do energy optimization and you get the ground state structures and you do energy calculations.
Yes.
Okay, so how do you know if what you have is a ground state structure or a transition state?
So I've been optimizing it specifically to the ground state. I actually have tried to get transition states, and it's been very difficult for me. So I've just been optimizing the energy to a minimum using the Gaussian 16 platform.
Yeah, how do you know if you have a minimum?
I initially ran multiple iterations, and once I had one that was consistently staying at the lowest at a particular energy, that's the one that I started working with.
Oh, I see. Did you do like a frequency calculation just to
Oh,
confirm?
yes, yes.
Okay, you did frequency calculation. Okay. All right, thank you.
Hi, Meg. I got a quick question. So I think overall you were talking about how this would help in future, I guess, cancer diagnosis by looking at differential sugars in or glycans. I'm just wondering, like, how easily are these summary statistics implemented to where you can look at them, like maybe in a clustering analysis? Have you thought about how your system might be implemented further on?
So right now, what we've done is essentially like looking at the effects of both the hydroxyl location, so primary versus secondary, and then sodium adduction in order to explain some of the fragmentation patterns that we've seen during CID in tandem MS. So ultimately, the goal is to be able to look at these parameters and say, you know, we have a primary hydroxyl here. We're seeing that on this sugar, this particular hydroxyl is exchanging most rapidly. Therefore, because we saw energetically that this site is more favorable, we're predicting that because we see labeling here, that means it's this type of hydroxyl or this type of site. So using that in order to better elucidate glycan structures later on.
Okay, I see where that's going then. Thanks.
Yeah, hi Meg, that was a really good presentation. So yeah, given I'm a little bit more familiar with Dr. Gallagher's research, yeah, I just want to know a little bit more about how are these how are these different conditions that you're introducing in your calculations, how are they replicated in the in the physical experiments with your instrumental setup?
So I'm purely computational. What I've really done, one of our grad students just graduated, and the work that I was doing was supporting a lot of what she had done. So she was seeing particular scrambling patterns in her investigations of HDX, and she wanted to better understand, why we were seeing this particular fragment, this particular scrambling. So we were looking at these energetics in order to say, here's where exchange is going to be most favorable based on computation, and then applying it to say, well, that's why we're seeing this particular fragment coming out when we're running CID on these sugars.
So I realize that you're only using sodium here, but perhaps what would you say about using other adducts to maybe provide further confirmation, just allowing, corroborating changes in your calculations with changes in your mass spectrometry experiments? Would that be reasonable?
Yeah, that's something that's been suggested. So there's always room for further study, for sure.
Yeah, perfect. All right, thank you.
All right, so thank you, Meg. You can go ahead and unshare the screen now.
Would normally take place. However, in a patient with ARDS, due to this fluid buildup, the lungs would be prevented from filling up with sufficient air, which will inhibit the gas exchange. And as a result, this can have downstream effects on the organs receiving sufficient oxygenated blood and potential organ failure. ARDS is often caused by another condition, most commonly sepsis or widespread infection of the bloodstream. However, most notably, it can also be caused by direct diseases pertaining to the lungs, such as pneumonia, or very pertinent now, COVID-19. In fact, 67 to 85% of COVID patients admitted to the ICU had ARDS, which emphasizes the need to study ARDS more in depth. So thus, due to this rise of COVID, as well as the fact that many of the patients that have ARDS are typically critically ill, as mentioned before, the disease can be quite fatal and is therefore of key interest for study. We had many research objectives in the creation of this database. First, we wanted to address a gap of knowledge, namely that prior to the creation of our database, there was no freely available database solely dedicated to ARDS that provided metadata and basic gene level information from experimentally verifiable genome sequence studies. Furthermore, we set out to accomplish 4 particular goals. First, we wished to develop and deploy a database for use by the scientific community using the NIH FAIR standards for resource development, and I will expand upon the FAIR standards later in the presentation. Second, we wish to facilitate the ease of studies exploring ARDS gene associations. Third, we hoped to highlight important metadata from experimentally verified studies. And fourth, we hope to provide a framework for future machine learning research pertaining to ARDS. So here is a general workflow of our database curation process. So first, starting with these 2 steps on the left, we sought to search NCBI and PubMed for research papers related to ARDS. And based on our process, we hoped to first kind of start with the review papers that were more broad, and then from there move to this step in the white box, namely to see the cited papers to find more specific experimental studies that could provide additional information to the genes. From there, we would go to this step, namely to use NCBI's Genome Data Viewer, which would in turn provide us with the relevant data to fill in our database, such as obtaining the chromosome location, obtaining the base pair location, and so on. These two steps in green, I'll touch upon later, but these were essentially a detour step that would eventually get us back to this red step here, in which we would follow the steps in green when a gene was found in a paper under an alias name and we needed to convert it to its official gene symbol. So now I will kind of outline the process using a sample gene, in this case, EGLN1, which was one of the 239 genes in our final database. So here you can see that we would do just say a general search on NCBI for relevant research papers. And here the search was ARDS genomics. And this first link gave us a table of genes that were found to be linked to ARDS, including EGLN1. And now I will explain what we would do if we found that a gene was under an alias name. Namely, we would use something called gene cards. So here's a screenshot of the gene cards interface. So in some papers that we would look, we might find the gene under an alias name. And in this case, some alias names could be HALAH or SM20. And here, if we were to search it into the gene cards, we could then track back to finding the official gene symbol, in this case, EGLN1, so that we could have this official gene symbol for our database. From there, as mentioned before, we proceeded to this key step, namely to use NCBI's Genome Data Viewer to find the detailed metadata that we wanted to include in our database. Here is an image of what the NCBI Genome Data Viewer would look like upon doing the search. In this case, as mentioned before, EGLN1. And the key information that we wanted to take from the Genome Data Viewer was, after hovering over this green bar, this table would pop up, and we specifically wanted to use NCBI's Gene ID page and navigate to this page from the Genome Data Viewer. On this Gene ID page, we would be able to obtain the relevant information. And the relevant information included, as mentioned on the workflow diagram, things such as the official gene symbol and the full gene name. We could also find the summary of the gene function, which we later included in our database. Also, you see some of the alias names that were also shown on the gene cards, such as SM20, HALAH. Also, we could obtain the cytogenetic or chromosome location. And just to help understand this 1q42.2, that refers to the fact that it was on chromosome 1, on the Q or longer arm, region 4, band 2, and subband 2. And furthermore, another thing that we could obtain from this page was the base pair start and stop location from the Genome Reference Consortium, given these numbers here, which we also chose to include in our database. So now zooming out, here is an image of the first part of the entire database entry for the gene EGLN1. So as mentioned earlier, we could obtain the official gene symbol, full name, and alias name, gene ID, the cytogenetic location, and the position start and end from the Genome Data Viewer process, as mentioned before. These 5 columns pertain to the variant that could be linked to ARDS, although this was a much more specific thing to particular genes and didn't apply to all genes. And I would be happy to go into this more in the questions section, but basically this was obtained through further experimental research on experimental studies that might mention a particular variant that was linked to ARDS. Now, moving on to the second part, we have the references and the primary sources. The references was referring particularly to the review papers that we found that would mention the gene. And so in this case, we have the PubMed ID with its external link so that researchers could then see where we found the information that pertained to the gene. And this PubMed ID actually would take you to the link that I showed in the first stage of finding the relevant genes. The primary sources referred more specifically to the relevant cited papers that the review paper might have cited to provide us additional information with regards to the specific link between the gene and its relation to ARDS. Here, the gene function, as I mentioned earlier, was obtained from the genome data viewer process. And these final two columns, with regards to the mortality or susceptibility and the particular relationship between the gene and ARDS, could be found from either the review paper or potentially a specific experimental study that was cited, if applicable. And now here is a version that you might see as a user of the database if you were to go to Zenodo and download the .db file of the database. And here you can see the first 20 rows, including EGLN1 at the top. And yes, this just helps to get a view of what the user might see when they would want to download it off of Zenodo. And as mentioned earlier, one of our primary goals was falling under the FAIR, NIH FAIR principles. F referring to findable, such that the entries would be able to be located quickly and efficiently. A, accessible, that the data would be open, free, and universally available. I, interoperable, such that there would be a common programming language for broad applications, in this case, SQL, which we just got from exporting our Excel to the SQL. And then reusable, namely that the data could be clearly described for others to use in the future. Now, conclusions. In conclusion, we are happy to say that we created a freely available ARDS gene database with 239 associated genes with regards to ARDS in our first version. And we hope that this may provide the foundations for others to use and develop upon in their own research. And also we hope that it may provide the framework for future machine learning applications, as I would be happy to explain in greater detail in questions. But in short, just as an example, a teammate and I are currently working on using random forest algorithms to further investigate the links between the ARDS genes that we found in our database and patient data sets. In addition, we also have hopes for to create a second version, in that we may have a friendlier web portal with better visualization options to help future researchers with ease of access in their data analysis and data research. Here are the references, and I would lastly like to thank my teammates, Eric Quintanilla, Fajar Adnan, Kimberly Dewa, Ashley Nguyen, Lavong Vu, Mary Claire Altruz, and our principal investigator, Dr. Ina Marie Toby. And thank you for listening. I'm now open for questions.
Hi, Samuel. I got a quick question. So where you talked about how particular variants are linked to ARDS and how you're keeping track of those, I'm just wondering how strong of a link should it have? Should it be correlative or regulatory in nature?
So for the purpose of this database, we were willing to go more on a correlative aspect based on what we would find in the relevant literature. I think in the future, it might be of interest to kind of investigate regulatory impacts, but this database was focused kind of on a correlative nature to begin with.
Great, thank you.
Hi, Summer. I
Go ahead,
guess,
Patrick.
okay, thanks. I guess probably because your work has to do with getting database that people can use in future and all that, because some of your slides, it was difficult for me to see what you're saying. But I guess like with database, you have lots of information in one slide, and so it's just difficult to like pick out what you're saying in particular slides. So my question is, you said you guys are developing the machine learning framework. Would you guys also like do the algorithm and like do the machine learning phase of it, or you just kind of like get the framework and stop there?
Yes, we would hope to do the machine learning part. So actually, yes, this is just kind of a basic outline, but I think in our current, me and a teammate and I, we are currently trying to use random forest algorithms such that we could kind of use various, kind of have various genes from our database serve as the nodes of the random forest algorithm such that we could kind of further validate or come up with new genes that could be linked to ARDS given a patient data set. And so we're currently working with a patient data set with a group of patients with acute lung injury and sepsis. And acute lung injury is kind of a broader demographic that would include ARDS patients and then those without. And so we kind of hoped that the database would provide the kind of the starting foundations to at least give us a starting point to find the link in an actual patient data set, just because, of course, in say with microarray data, there would be say like thousands of genes, 20,000 or so, but it's kind of hard to just go in starting with like 20,000 candidate genes. So we hope that the gene database can kind of has allowed us to give us a starting place in terms of limiting our scope, at least to begin with.
Yeah, because it would be good to see like the training set you guys develop, and also just like you said, if you guys have a patient, to see if you're going to get if you can detect things like this from your algorithm.
Right. Yes, that would definitely be in store for future directions of research. So I definitely appreciate the recommendation.
Thank you. Luke, you can go ahead.
Thanks. Yeah, and sorry if I missed this. Was your database curation or generation curation, was that a purely automated process, or is there a mixture of automation and manual curation?
So actually, this one was much more so manual. So we kind of just had a large team of undergraduates that did it in a more manual step. So actually, our main and this would definitely be a room for improvement, I think, in terms of trying to be comprehensive in that like there could be ways to further streamline the process in an automated way, but we kind of went on just more of a manual way with lots of hands. And when we found that references were starting to reference each other and we were just kind of finding the same
Going
papers
circles.
over, right, then we kind of at least deemed it sufficient for the first version. But of course, I guess that's why we also wanted to make it updatable so that it can be further improved.
Okay. Yeah, I guess I was wondering, because when you're manually curating these types of things, you know, you're more at liberty to make, you know, decisions about what you include, what you don't include. And maybe this was touched on in an earlier question, but is a mention of a gene set in a paper sufficient criteria for inclusion? What criteria does a gene have to meet to be included?
So for this set, for this version, it a mention usually would fall in kind of like a table, say for a review paper, and that was usually sufficient grounds. Obviously, some of those would have, I guess, fewer entries or maybe less detailed information in kind of the details section, but at least for kind of raw inclusion, we were willing to include I guess a raw mention, usually in the form of, say, a table in a review paper.
Okay, thank you.
All right. Thank you, judges, and thank you, Samuel. Our next presenter is going to be Tuba from Baylor University. So whenever you're ready, you can go ahead and share your screen and start.
I'm going to get out of presenter view really quick and then I will start.
No problem.
I don't know, if you're using two screens, there should be like a swap button at the top that you can just click on and to show the other one.
I'm only using one screen, so I'm not 100% sure. So it won't let me swap because I'm using one screen.
Oh, then maybe you just go with slideshow instead of presenter's view. I don't know.
Okay. Can everybody just see the slideshow now?
Yes.
Okay, I'll start. Hi, my name is Tiba Harris. I'm in the Benton Lab at Baylor University. For our research, we are identifying patterns of transcription factors binding motifs in a combination of enhancers. So in the Benton Lab, we are trying to find evidence of redundant enhancers, and quantifying the similarity between different enhancer sequences will help us better understand how enhancers cooperate to regulate genes. It can also help us understand the effects of regulatory genetic variation in liver cells. Essentially, enhancers are gene regulatory elements that control the transcription of genes. They help with proper growth and development of cells and tissues, and they contain binding sites for transcription factors. So enhancers play an important role in gene expression. They activate transcription of genes, and mutations in these regions can cause an appearance of traits that are likely to participate in disease processes. So in the first image, it is an example of preaxial polydactyly, which occurs with various point mutations in the ZRS enhancer. And this refers to polydactyly, where an additional digit grows towards the first digit of the hand or the foot, so near the thumb or the toe. And the second image, it is an example of a Pierre-Robin sequence, which occurs with deletions of SOX9 on chromosome 17. And it is a condition present at birth in which an infant has a smaller than normal jaw and a tongue that is placed further back than normal. And there is an opening at the roof of the mouth. So in this image, we can see that the enhancers are bound by activating transcription factors and are brought to close proximity of their respective target promoters by looping, as we can see in part B. And in part C, we can see that these element functions function at a distance by forming chromatin loops to bring the enhancer and the target gene closer into proximity. Studies have shown that combinations of enhancers can regulate the same gene and can have redundant elements. And these redundant enhancers can help reduce the effect of genetic variants on gene expression levels. And in this picture, you can see that the shared and separated transcription factor inputs to the individual enhancers have different effects on gene expression noise. Lower expression noise is seen with shadow enhancers with separated transcription factor inputs. And shadow enhancers are similar to redundant enhancers as they present redundant functions. And there are groups of 2 or more enhancers that control the same gene target. Here we can see a schematic depiction of an enhancer and a target gene, and it is marked with biochemical annotations used to nominate candidate enhancers and other features of non-coding DNA. If you look at the transcription binding motifs, those are especially involved in transcription regulation. So in our research, we are analyzing a set of 70,000 human enhancer sequences in liver cells with predicted gene targets to find evidence of redundant enhancers. We are looking at similarity of patterns of transcription factor binding motifs within enhancers within the same target gene. So we used 2 methods. So one of the first methods we used was scanning enhancer sequences and putting them in a data frame. So we created a data frame of all the motifs present in different enhancer sequences, and we found over 400 different motifs and over 70,000 enhancer sequences. So this is our data frame. So the first column of our data frame shows the sequence name. It tells us which chromosome it's part of, the start and ending location of the enhancer sequence, and every column after that is an enhancer sequence, is a motif present or not. So we have the ARH and the AIRE and so many more. So the numbers in this data frame tell us if there is a enhancer present, and if it is, and how many counts of that motif are present in that enhancer. So with a bunch of these zeros, we can see that these motifs are not present in those enhancer sequences. But with the 3, we see that the AP2A motif is present 3 times in this specific enhancer sequence. Towards the end of our data frame, we have a more detailed version, which reinstates that which chromosome the enhancer is part of, the start and end location, and it also tells us what gene, what target gene we are looking at, and the group it is part of. And the group comes into play whenever we analyze the data further. So the top 10 most prevalent motifs in our data frame, we made a bar graph out of them. And as you can see, the motifs are on the x-axis, and on the y-axis, we have the total count, the sum of the motifs that were present. And you can see that MAZ we can see a depiction of how much variation is in between these motifs. So we found MAZ to have over 2 million hit, and it was our most common motif present. And VEZF1 and ZN467 have approximately over 7 million hits. So the most common motif was MAZ, and MAZ participates in transcription termination and polyadenation, which adds adenines at the end of RNA to stop transcription. It also deregulated expression of MAZ is reported to correlate with various tissue malignancies. And the sequence right here is a sequence logo of MAZ, and it is a graphical representation of the sequence conservations of the nucleotides found in this specific motif. So the second most common motif was VEZF1, which is involved in normal and abnormal cell proliferation and differentiation. And the third most common motif that we found was ZN467, and it is a transcription factor for several genes, and it can help recruit histone deacetylase complexes, which remove acetyl groups from histones, and it tightens the chromatin structure, and that can help terminate transcription or prevent it from happening at all. So the second method we used to analyze our data was principal component analysis. So principal component analysis, or PCA, is a dimensionality reduction method that is often used to reduce large data sets and by transforming a large set of variables into smaller ones, but that still contain most of the information from the large data sets. So I guess like in simpler terms, PCA is used to reduce the number of variables of a large data set, but while still preserving as much information as possible. So we go from a data set like the previous ones that I showed you to something similar to a scatter plot. So first we created a PCA data frame, and we used the sklearn library in pandas to calculate our PCA. So after calculating that, it gave us PC1 and PC2. And PC1, or first principal component, is the direction in space along which projections or the data points have the largest variance. And the second principal component, or PC2, is the direction which maximizes the variance amongst all directions. And we're expecting to find possible evidence of redundant enhancers, and we're looking for clustering of enhancer groups based on transcription factor binding. So this is an example of our data. So this is PCA data of all of our enhancer groups, all 70,000 of them. And we can see it's really dense in just like this specific area right here. So we tried to group our data into the groups. We can analyze that better, but we still had that dense area right there, and we couldn't visually see that much of all the points in our data frame. So we try to isolate our data using a specific chromosomes. So this graph right here is a PCA data for specifically chromosome 21. And we can see we still have that trend of having a dense in towards the left, but but it's easier to visualize the data if we isolate them through the chromosomes that they are a part of. So our hypothesis was to use PCA, or principal component analysis, to separate observations in two-dimensional space to visualize things with similar values of transcription factor binding sites. And our goal was to look for clustering of enhancer groups based on the transcription factor binding sites, and to reduce the huge data frame that we originally had to something that we can interpret. So we scanned approximately 70,000 liver enhancer sequences for motifs, and we quantified the number of transcription factor binding motifs in each of these enhancers. So we started using dimensionality reduction techniques on binding site profiles, and in the future we are going to continue to explore this PCA and continue to look for ways to calculate similarity between groups of enhancers. Thank you so much for your time, and I'm open for any questions.
Hi, Tuba. I got a quick question. So you talk about your data frame having lots of zeros, and I think with your principal component analysis, you used a covariant matrix, correct?
Yes.
Okay. So have you tried any other types of dimensionality reduction that would be a bit more conducive to your data, which is like, it seems like it's probably more Poissonian distribution than it would be normal. So I'm just curious if you've tried like an NMDS or anything like that.
We haven't tried any other reduction methods so far, but that is something that we're looking into, because our PCA is looking very clustered in a specific spot, and it's not letting us visualize the data or analyze the evidence of redundant enhancers. So that is something that we're looking to add to our research.
Thank you.
Hi, Tuba. So what information, sorry if you already touched on this, what information can you derive at this point from your PCA analyses?
Yes, so we saw that it was really clustered in the left side of our data of our graph. It was very dense, and looking at that, we can we can elucidate so far that there is evidence of redundant enhancers, because we saw that specific groups, for groups especially under 2,500, were specific to that specific area. And that was showing evidence of redundant enhancers. And using that, we can see if if that really implements into gene expression noise.
Thank you.
Too bad. That was a good presentation. So mine is more of just foundation question. What is principal component analysis and how does it work?
Okay, so principal component analysis is a reduction method, basically, because we have such a huge data frame. We're trying to reduce that data frame into something that we can interpret. So using like the sklearn library that we use for our data frame, we are calculating the PC1 and PC2, and we're looking, and that's reducing from like our huge data frame to basically a scatter plot. So it shows us clustering, which we can use to like find evidence of redundant enhancers and possible feature effects.
How do you decide on your principal components? How do you decide on which should be one and which should be two?
So it's not something that we decide. Whenever we use the PCA library that's part of sklearn, it calculates it for us using computational methods.
Okay, just based on correlation, I guess, like the one that is most pronounced followed by the other one. Okay, thank you.
Thank you.
All right, thank you, Tuba. You can go ahead and unshare your presentation now. So thank you to all the presenters. You guys all did a great job. And this concludes the Bioinformatics and Computational Chemistry division.